Hey class, here's a slightly more advanced version of meiosis. Okay, so I'm going to draw a cell, a germ cell. Again, this would be a progenitor for a sperm. And this germ cell, again, has it has a pair of chromosomes, a single chromosome. So again, we are sexually reproducing organisms, so we've got our chromosome contribution, one from mom, one from dad. So I'm going to color code. I'm going to take advantage of the color coding of the app. So the first thing that has to happen, again, in meiosis is the replication of DNA. This is happening. Oops. Get rid of that. This is happening prior to the cell division event, okay? So we'll draw slightly larger so you see what's happening here. Okay, so you've got DNA replication, which gives you sister chromatids. Okay. There you go. There, the pairs of homologous chromosomes, which is a pair of sister chromatids. Okay. Now, here's the thing that makes this advanced, sort of the advanced version of meiosis. You're going to have something happen here, which is known as crossing over, which is going to give you an additional bit of variability. And crossing over is going to look as follows. Okay. So I'm going to draw this again. I'm going to draw the chromosomes in a slightly special way. So I'm going to draw one chromosome looking like that, and I'm going to draw the other pair of chromosomes looking like this. Okay, so let's just assume that this crossing over event occurred, and the consequence of this crossing over event is that you end up with your exchange, your recombination of a bit of genetic material as a result of crossing over. Okay, so now what you've done is you've exchanged a bit of that chromosome from one pair of homologous chromosomes with the other. Okay, now when these go through the division cycle, so I'm just going to have to draw them off to the side. Okay, so these are going to divide into two daughter cells. Okay, and now the makeup of the daughter cells is going to look as follows. Okay, and again, I'm using the colors to help track the location of every bit of chromosome. So there you have, these are the two daughter cells of the first meiotic division. Okay, and because of the crossing over event, you're now taking some bit of chromosome that was in one homologous pair of chromosomes. You're going to take it along with the other. So you've exchanged a bit of chromosome material. And there's one more division in meiosis. Okay, so here we have it. I'm going to draw all the cells here. Okay, and again, these are sperm cells. I'm going to draw just sort of the flagella. There we go. And here's what the chromosome there we go. It's going to look like for each of these. All right. So now you've got. So if you think of the previous video, the simple meiosis video, you had 4 versions, okay, and 2 of them were kind of identical. But then you've got actually more versions of more possible combinations of alleles of genes, because you've got this exchange of chromosome material during the first part of meiosis, okay, because of crossing over. All right. So this is how you get enhanced variability as a result of meiosis. You get these crossing over events that give you more chances at having different combinations of alleles in the same chromosomes, and you wouldn't expect regularly. Okay, so I'm going to upload this too, and hopefully you get to download it and check it out.